We all know that cell-based models are still one of the best valuable tools during the drug discovery stage and very useful for preclinical testing because it's easy to use and can do high throughput screening easily. So now I'm going to take you to a short journey to search for the best in vitro kidney cell toxicity models. So for those cell-based models, what we are looking for is high predictability, has a mature kidney proximal tubular origin, and best mimics the proxitubular microenvironment. Let's start with the primary cells. As Kevin has pointed out, the primary cells can make excellent in vitro models because they often best represent the in vivo situation. However, in the case of kidney cells, the primary RPTEC cells quickly lose the expression of the three major important transporters such as OAT1, OAT2, and OAT3, and therefore are not relevant models in this particular case. Moving next to the continuous renal cell lines. For many years, there have been a number of renal cell lines available from a variety of species. However, even after all this time, none of these has been fully characterized to show that they are fully recapitulate the function of proximal tubular cells in vivo. The next step in the evolution of renal models are to use a gene modification method to overexpress transport genes in either long kidney such as U2S, a CHOS cell, or embryonic kidney cells such as HK293T cells. ATCC has a version of these first generation models that overexpress the OAT1 as shown on the left hand side. However, these models still lack the critical proximal tubular microenvironment characteristic. To address this and develop more physiological relevant next generation renal models, my lab started with an immortalized proximal tubular cell line and then stably integrated SLC transporters into this cell. I'm now going to walk you through the generation and the application of these cell lines. So let's first look at the parent line, where our models are based upon the immortalized human adult proximal tubular cells. This cell is derived from a normal renal proximal tubular epithelial and immortalized by H3 only. So these cells have been fully characterized the uniform expression of E category and CD13, which is a resident renal protein. Importantly, it forms dome-like structures. So domes are multicellular cystic structures which have been described previously in epithelial cultured from various tissues that have a long transport or secretory function in vivo and in vitro. From here, you can see these cultured cells recapture the characteristic of the in vivo proximal tubular characteristic. And also, these cultured cells have stabilized TIA characteristic. It's called trans-epithelial electronic resistance. So all these characteristics are best represented the in vivo micro proximal tubular characteristic. So any models based on this cell line would be best capture the characteristic of the in vivo situation. But unfortunately, these RPTEC 31 cells, like its primary counterparts, lost the OAT1, OAT2, and OAT3 expression in culture also. So our strategy is to overexpress the OAT1, OAT2, and OAT3, these three most important SLC transporters recommended by the government agency back into the RPTEC 31 cells. And we choose the best clones to begin with to characterize the cells. First is the molecular characterization of these OAT1 clones. You can see we did figure A is the mRNA level by RT-PCR from different clones The figure B is the protein expression from these clones. And the figure C is the sequencing of one of the clones that show there is no mutation. And also we did a ddPCR, verified the copy numbers of the OAT1 incorporated in the chromosome in this clone are 7 versus 2 copies in the parental line. One thing we need to point out is either in figure A or figure B, the RPTEC, the parental line, does not express the OAT1 at either an mRNA or protein level. So this slide verifies the OAT1 is correctly expressed in the clonal cell lines. And then next, we want to look at the overexpressed OAT1, the clonal cell line. The OAT1 can correctly localize to where it should be. So from this figure, the red color is to show the subcellular localization of the overexpressed OAT1 is exclusively localized cell membrane. And the lower panel will show the parental line. There is no expression of OAT1. It's also match our bimolecular characterization. Secondly, we characterize the OAT2. We also choose some clones, verified the mRNA level in those clones by RT-PCR, and Western blot for the protein expression in those clones. And figure C is the sequencing data showing that these cells, the OAT2 has no mutation. We also did a ddPCR, verified the numbers of copies of OAT2 in those clonal lines. One of the clonal lines is 5 versus 2 in the parental line. Another thing also need to point out is that the parental line has no detectable either mRNA or protein level of the OAT2. And here, this figure also showing the overexpressed OAT2, the localized subcellular localization. It's correctly localized to the cell membrane compared to the lower panel, the parental line, which has no OAT2 expression at all. Thirdly, our coming soon, the OAT3 cell lines, we also did a characterization. The first by RT-PCR to verify the mRNA expression. Then on the right hand, the ICC, the top 2 figures showing you the membrane localization of the OAT3 compared to the parental line has no detectable OAT3 expression. And we also verified the copy numbers of OAT3 has a 5 versus 2 in the parental line. So from the above 4 slides, we know that the overexpressed OAT1, OAT2, and OAT3 in the molecular biochemistry and subcellular localization are correct. Now we will ask how biologically they are similar to the parental line. First, let's look at the morphology. You can see from we listed the cells at a low density, high density, that looks very similar to its parental line, which is the first 2 pictures. They looks very similar. Secondly, as we have been talking about before, we want to investigate how those domes are formed in the clonal line. You can see the picture show here. You can see the parental OAT1, OAT2, and OAT3 clonal lines all can form the domes very efficiently. So these are very important features to recapture the in vivo situation. And also we stain the several important markers, the CD13, as well as the E category for those clonal lines, OAT1, OAT2 versus the parental line, as well OAT3 versus its parental line. So they show similar expression of all the markers.